Hello everyone! Welcome to today's live broadcast, Humans and Fish to Study Cellular Mechanisms of Neuromuscular Development. I am Kristen Perechentil of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. For more information, please visit labroots.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button, lower left. This is an educational web seminar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your web page and follow the process of obtaining your credits. I would now like to introduce today's speaker, Dr. Chiara Manzini. Dr. Manzini is a human geneticist and cell biologist whose laboratory focuses on the molecular mechanisms of brain development. Her group at the George Washington University identified disease-causing mutations in families affected by brain malformations, intellectual disability, and autism. She uses both zebrafish and mouse models to study the cellular function of these disease genes. She obtained her undergraduate degree in molecular biology from the University of Pavia in Italy, a PhD in neurobiology and behavior from Columbia University in New York City, and completed postdoctoral training in neurogenetics at Boston Children's Hospital and Harvard Medical School in Boston. She joined the GWU faculty in 2013. I will now turn it over to Dr. Manzini for her presentation.
Hello, thank you, Kristen, for the introduction, and thank you, LabRoots, for giving me the chance to tell everyone about what my lab is doing. As Kristen said, we do use a combination of human genetics and animal models to study the mechanisms of neurodevelopmental disorders. And today, in particular, I will tell you about a project focusing on neuromuscular development using zebrafish as a model. So what is the big question? The big question we're trying to understand is how does the brain develop and how it goes from a small vesicle really early during human gestation to this beautiful, complicated organ that controls everything that we do in our lives. What my laboratory is trying to figure out is what happens when there's an insult during development that affects the developmental processes regulating the brain, causing either structural or functional deficits. Structural deficits are brain malformations where the brain structure will be disrupted. And functional deficit could have a relatively normal-looking brain but still have deficient functions such as an intellectual disability and autism spectrum disorder where cognitive function and social function are affected. So how do we do this? We start from humans, and so we identify families that have specific developmental disorders that affect the brain and do genetic studies to identify the genes that are responsible. Then, once that we've identified these genes, which we know to be essential for human development, we try to figure out why they're being perturbed causes disease. With two principal goals. One goal, of course, is to better understand the phenotype of the patients. And so to understand what is happening and how the different organs that are affected and how brain development is disrupted. But also, if these genes disrupt brain development, they must be incredibly important for that process. And so we really want to also define the molecular mechanisms that they control, and that is a more essential, more basic function. What I will tell you about today is one of the projects in the lab that focuses on the role of the extracellular matrix in the differentiation of neurons. It's a cool project because it uses both human genetics, so next generation sequencing, which are state-of-the-art sequencing technologies, and in vivo validation of identified genes in zebrafish, and to basically figure out where the patient phenotype is originating from. At the beginning of this project was led by multiple students and techs in my lab, Stefania Di Costanzo, who has now left, and she's in graduate school at UCSD, Heather Pond, which manages our zebrafish facility and is still in the lab, and a very talented undergraduate, Habon Issa, who has also moved on. So why the extracellular matrix? So the extracellular matrix is a very interesting structure. If this is the plasma membrane and the cell membrane, the extracellular matrix is outside the cell, and it is a meshwork of proteins and polysaccharides. Polysaccharides are like chains of sugars that are also called glycans. And this structure is very complex. It's very difficult to tease out what all the different components are. And it was originally thought to pretty much act as a glue. So these are cardiomyocytes. It's a really beautiful electron microscopy image. And the extracellular matrix is supposed to hold, is holding the different muscle fibers in the heart together, acting kind of like as a glue. And that was originally the idea of what the extracellular matrix was doing. Now we know that things are a lot more complex. The extracellular matrix, the ECM, surrounds neurons throughout development, and I became interested in it actually when I was a graduate student, and I started doing experiments on it. And so here you can see these are neuronal progenitors from the cerebellum with a stain that shows that there is extracellular matrix surrounding all of these cells. If you look at the section of the mouse brain, you see a very, very strong layer of matrix at the very top. This is the basal lamina, which is very important, we will see, for neuronal migration and for brain integrity. But it's also extracellular matrix distributed across different layer of the cortex. This is a particular kind of proteoglycan. And so this differential expression at different stages of development and different localizations pointed to the fact that the ECM also has a functional role for neural differentiation that we're actually trying to figure out. So if the ECM is disrupted, another support for the fact that the ECM is important for brain development is the fact that if ECM is disrupted, you can have multiple types of brain disorders. So here I am listing, I'm showing some laminins and collagens, transmembrane proteins such as integrins and dystroglycan, and the 7 transmembrane domain, G protein receptor GPR56, and mutations in all of these genes that are indicated here cause disruptions in brain development. And so you will have a normal brain. If you look at the bottom, you'll have a normal brain on the left, and then disruptions of white matter with the laminin alpha-2 mutations, disruption in organization of the brain with GPR56, and very severe disruptions, ventricular enlargement, disruptions in the cortex with the alpha-dystroglycanopathies, which are defects in dystroglycan. So these transmembrane proteins are often glycosylated, and as I said, they have short sugar sequences that regulates their interactions with each other and control how they form, how the extracellular matrix is formed. The protein we're particularly interested in is alpha-dystroglycan. Dystroglycan is a two-part, it's cleaved into a beta protein and an alpha protein, and the alpha protein is very heavily glycosylated. It interacts with the extracellular compartment, extracellular matrix component, such as laminin, through its glycans. When these interactions are disrupted, that is when you get disease. And so these disorders are called dystroglycanopathies, which is a very complicated name, and it basically means diseases caused by defects in dystroglycan. What happens in these disorders is actually across multiple organs, because dystroglycan has a very important function in multiple systems. One of the primary deficits that you see at the top is actually in the muscle. On the left-hand panel in the dystroglycan glycosylation, you see the dystroglycan is glycosylated around the muscle fibers. This is it's a human muscle biopsy. It's glycosylated, you can see the glycosylation around the muscle fibers, and it's missing in the patient biopsy almost completely. What that does, this leads to very severe muscular dystrophy that is usually present at birth, and so it's congenital, and it's one of the most severe forms of muscular dystrophy that have ever been identified. In addition to that, the loss of dystroglycan glycosylation also causes severe ocular defects in retinal development and also in the formation of the anterior chamber, which is the lens and the cornea in front of your eye, and very severe brain malformations. And here you see one of the most severe brain malformations, which is a cobblestone-less encephaly, where the cortex is pretty much smooth, the ventricles are incredibly enlarged. As you see, it's a completely empty space, but also the brainstem and the cerebellum are severely affected. In addition, you may have renal malformations. And so loss of dystroglycan glycosylation affects multiple tissues, and what this tells us that this process is important in various places in the body. So let's focus on the brain. When you look at a brain with cobblestone-less encephaly or the dystroglycanopathy, you see this very characteristic disruption that you see in the middle, where the white matter and the gray matter are intermixed. There isn't a clear definition, and that is a very severe neuronal migration deficit. And you'll see it also in the pathology on the right, where instead of having a nice 6-layer organization of the cortex, you have these clumps of cells that are sitting all over the place. And so just to recap on what happens when the cortex develops, you have a progenitor cell that extends from the ventricle to the pia that will generate new cells, new neurons. The neurons will migrate using the progenitor as a scaffold, and they will organize and laminate the cortex. In this particular condition, you have a disruption in your progenitor cell. You have a disruption in the basement membrane, which is that very thick layer of extracellular matrix that I was telling you about at the very beginning. And so the anchoring of the progenitor cells of the radial glia into this basement membrane is disrupted, and the neurons don't migrate correctly. Some of them over-migrate, so they'll go above on the surface of the brain, or they will stay below in the white matter. When they go above on the surface of the brain, they form clumps of cells, and actually this is what gives the name of the malformation, cobblestone-less encephaly, because when you look at one of these brains upon autopsy, you basically see little pebbles of cells sitting on top of the brain. So one of the problems with dystroglycanopathies is that, yes, there's this very, very severe form that I just told you about, but they can also cause a different range of phenotypes, as you can see here on the bottom. The genes that we have identified so far only explain 40 to 60% of the cases, and most of them regulate alpha-dystroglycan glycosylations, bringing us back to that mechanism that we were talking about at the beginning. The problem is that it's very hard to diagnose one of these patients because there's a lot of genes. We don't know whether one of the responsible genes is going to be involved in this particular case, and the presentation of the brain can vary. And so you often have to test every single one of the genes, which can be quite expensive, and you only have a 50% hit rate. So what do you do when you have to deal with a heterogeneous disorder? A lot of groups have focused on families that are more genetically informative, so that you don't need multiple families with the same disease, but you can actually just focus on one individual family to try and find out what is wrong with them, which will help you with bypassing some of the heterogeneity. And so to do that, you go to countries where there is a higher incidence of consanguineous unions, which means first, second, or third cousin marriages. And this is a map of where consanguineous marriages happen around the world. They're most frequent around the world, and the blue asterisks are where some of our major collaborators are located and where our families are collected. So why do we do this? The reason why we do this is that studying a family which is consanguineous, like this first cousin union, allows us to use a genetic trick. So when you have a consanguineous family, you expect the mutation to have originated in the past from one of the ancestors and would be this like little cross on the orange chromosome. As the mutation passes along through the different generations, at some point you will have two descendants of the person with the original mutation that both carry that chromosome they inherited from their ancestor. That one chromosome is identical, in like that one DNA region is identical, and it will meet itself, and it will be homozygous, which means that it will be the same region inherited from the mom and the dad from a common ancestor. And that will cause the disease in one of the kids. And so what you do, you focus on the regions of homozygosity. This is very helpful because in a first cousin union like this, around 6% of the genome is predicted to be homozygous, and so you can actually remove the remaining 94%. So let's see how we do this. So this is a family affected by Walker-Warburg syndrome, which is the most severe form of dystroglycanopathy. Has congenital muscular dystrophy and that very, very severe cobblestone leukoencephaly that I showed you at the beginning. They're a first cousin union, that we have only one child in the family who is deceased. And so we used a mapping method, which is called like SNP arrays, to map the homozygosity or heterozygosity across the entire genome. As you can see here in like gray, black, and darker gray, those are the chromosomes. And in blue above them are the regions of homozygosity. If I can draw your attention, for example, to chromosome 8, where most of the chromosome is homozygous. What we found in this family is that 9.6% of their genome was homozygous, which encompassed almost 2,500 genes. So it's a tenth of the genome, and so we can only focus on that. But of course, 2,500 genes is a large number of genes. We were lucky because when we were analyzing this family, that's exactly when next generation sequencing was coming online. Now it's a normal clinical test, but back at that time, we were some of the first groups to use it. And you could do whole-exome sequencing, which targets only the coding sequences of the genes. It's 1% of the genome. It's fast. It's cheap. It's very easy to do. And we expect that a large number of the disease mutations are localized in the coding regions. So when you do an exome, you get from 30,000 to sometimes 100,000 variants or more. This is just the normal variability that we have as humans. Most of it is common. Most of it is shared with people from the same ethnic background, with your family. And so what we needed to do was to identify a filtering strategy that would allow us to identify the disease-causing mutation. So we focused on the region of homozygosity because that's where we predict that our mutation is going to be. And so only on those 2,480 genes. Then the mutation has to be novel or rare. This is a very rare disorder, so it cannot be something that's floating around the population and that could cause disease in multiple people. We also expect it to be pathogenic, which means to disrupt the function of the protein. And so it can either be a missense mutation, a change in an amino acid, or a truncating mutation, which either causes a stop, a splicing change, a frame shift, something that will stop, that will prevent the full translation of the protein and will generate a truncated, non-functional protein. So we were very shocked at the very, very beginning because this filtering approach reduced the number of genes from almost 2,500 to 6 in this particular case. And as we've been doing this more and more with multiple other families, we find that on average, this approach reduces the number of candidates to 5, which is very exciting because you start from tens of thousands of genes that you can really only get to a very small number that you can count on your hand. Of all of the mutations, the 6 mutations that we identified in this family, one popped out because it was in a glycosyltransferase. There was a stop codon, so it was a very severe truncating mutation in a gene that's called GTDC2, which at the time we really didn't know what it did. But as we were sequencing other families with the same method, we identified 2 additional mutations and other truncating mutations. These are all homozygous, which means that there are both chromosomes, and they're, of course, because they were identified through homozygosity, another truncating mutation earlier in the gene, and then a missense change, which is very, very, very conserved across all of the different species. So we were very excited because we thought, okay, we have 3 independent mutations. This is really a great candidate. And immediately, looking at the small number of genes that we get, we decided that we really needed a validation plan that could be used with medium to high throughput to test all of these variants. This is where the zebrafish came into play. The zebrafish is great for genetic screening. It has really exploded in the past 5 years, basically, as a tool for studying genetic mutations because it develops very quickly and it has similar organs to us. And so you can actually recapitulate the disease sometimes in the order of a day or 2 days. It has very prominent eyes and muscle. If 1 hour, it's 4 cells. At 24 hours, it's this little larva. And you can see at the bottom of the eyes, the brain, and the tail, where the muscle is developing. It's fertilized externally, so you can take your eggs at a 1-cell stage, so we only have 1 cell before it divides, and you can inject it with multiple reagents to be able to disrupt your gene of interest. In this case, I'm showing you morpholino oligonucleotides, but of course now you can also do CRISPRs where you modify the genome of the very first cell to obtain a mutant fish. So what do we see? We see, first of all, you can start looking at the embryo in the chorion. The movement starts around 20 hours, and sorry. Nope. Okay, so it's not running a movie. So what I was going to show you here was a short movie where you see the spontaneous movement of a zebrafish into the chorion. The little zebrafish will flip its tail. It's doing exactly the same thing that babies do in the womb. It's activating, it's connecting its peripheral nervous system to the muscles. It's activating all of these processes, and it's moving. And it's moving all over the chorion. We can trace this particular movement, and so we can trace the head and we can trace the tail. And as you can see here, when you do the tracing, it travels around quite a bit. We then can video the fish that have been, where the genes of interest for muscular dystrophy have been removed, and I will skip directly to the analysis, and instead what they do, they'll move back and forth. They will not be able to generate enough strength in their tail to actually travel around and to do their tail flips. So that's already a change in their motility during development. What this does is actually that when they're born, as you can see here, they are born with a curved tail. This is very similar to what happens to the human babies, which are born with what is called arthrogryposis or contractures, which means that as they're in the womb and their joints cannot move, they're actually born with joints stuck in position. And one example that you might know that is common is like the club foot, where the ankle is permanently twisted. In this particular case, we can actually say like the little zebrafish has a club tail, so it's completely stuck in that situation. It cannot extend it. In addition to that, they also have very severe, so this is where GTDC2 was removed. They have very severe defects in the development of their head. You can see the retina is not fully fused and developed in the eye of this little fish. The shape of their head is not normal. You can definitely see a edema and a stroke where there is blood in the brain. So they were clearly not normal. The issue is, does this recapitulate what we see with the patients? And so we started, first of all, looking at the muscular dystrophy. The muscular dystrophy is one of the primary diagnostic criteria for this disorder, where you see loss in dystroglycan glycosylation. And so on the left, here you see the human biopsy with normal dystroglycan and glycosylation and laminin alpha-2, and the loss of glycosylation and disruption in laminin. On the left, you see the fish muscle. In the fish muscle, the extracellular matrix that you see distributed around the myofibers is actually distributed in these lines, in these longitudinal lines, the green lines that you see here, where we look at dystrophin and alpha-dystroglycan. And you see that in the morphin fish, where GTDC2 has been removed, you have a loss of both dystrophin and alpha-dystroglycan. So it's a very similar mechanism to the one, very similar defect to the one that we see in the humans. We were also really excited to see a similar brain malformation, because if you prevent pigmentation, you can actually see this extreme enlargement of the ventricles in the brain of the fish at 3 days post-fertilization. So this is just a 3-day-old embryo that's turned into a larva, so it's starting to swim. As you see in a section here in the organization of the brain, the ventricle of the zebrafish forms differently from the human ventricle because it fuses at the top. And so instead of having this very extreme extension that you see in the human brain, you see a compression of all of the brain tissue towards the ventral part of the fish. But it's very similar, and we can basically see that they also have hydrocephaly. In addition, we can recapitulate the retinal deficits. The zebrafish retina has the same organization as the human retina. They are very, very good. They see very well in color, so they're similar to us in that extent. And they have pretty much the same type of cells and laminar organization. One of the characteristics of the mutation of the malformation, the ocular malformation, is retinal dysplasia, so it's a disruption in retinal organization, and you can see here in the morphine, again, disruptions in retinal organization. The cells are much less compact, even in the ventral portion of the retina. So we are very excited because we could recapitulate all of the human phenotypes in the muscle, in the brain, and in the eye. Then the great thing that you can do into the zebrafish is you can start doing manipulations. And so you can use the human mRNA to see if you can rescue the morphin phenotype. Why is this important? It's important to show that the gene's function is conserved across evolution, and so that you're actually testing a function that is true in the humans. And so what you see here, we see that we use the human GTDC2 to rescue the phenotype in this fish. But the other thing that you can do, which is really interesting, is that you can take your missense mutations, because if you have a truncating mutation, sometimes you think, okay, the function of the protein is disrupted, but you can take a missense mutation, you can do the same experiment, and you can demonstrate that that missense mutation completely removes the function of the protein. And this is what we did with that missense mutation in GTDC2. So it's a really cool model also for studying mutation function. So this was great, and we basically started expanding this method and trying to use it in different ways. And so what was very exciting is that we were actually also able to look at families that were non-consanguineous, because of course you will have access to families from all over the world. In this particular case, there were unrelated parents from South Africa that had 3 children with Walker-Warburg syndrome. When we looked, we did the exome of both of the children that we had available, and what was amazing is that there was only one gene that came out that was inherited in one mutation from the mom, one mutation from the dad, and having 2 mutations in the kids. And this gene was called B3-galin2. If you are not a glycosylation expert, this name will not say anything to you. But if you are, you become really excited because it is a glycosyltransferase, and which is also involved in dystroglycan glycosylation. At the same time, we collaborated with other groups, Francesco Montoni, University College London, identified additional families in the same genes, plus in different families in the UK. And so we basically came up with a variety of mutations across this gene, causing the disorder. So that was very exciting, and the B3-galin2 morphin fish was identical to the GTDC2, so the zebrafish again confirmed that this is the phenotype that you can see. This can also be used to study variability. Another gene we looked at was called POMK. We had this one family with a somewhat different presentation. They had limb-girdle muscular dystrophy, which is type of muscular dystrophy, which shows up a little bit later, with reduced intellectual functioning. They are differently from most of our other patients. They were still alive. They were ambulatory, which means that they could do stairs and walk around in their 20s, so it was much less severe. And also a family with a child with Walker-Warburg syndrome, that very, very severe disorder. And in both cases, we identified alleles in POMK. POMK, differently from the other genes, is a kinase. And I'll tell you in a second what it does. And what was interesting was that family 1 had a homozygous stop mutation that basically removed the entire kinase domain, while family 2 had 2 heterozygous mutations, which one was a very early frame shift and the other one was later in the protein. And so we were wondering why the loss of the entire kinase domain, so that caused this disorder, was not as severe as this particular case. And so we developed a POMK morphin, so with the morpholino to look at the development of the fish. This is not as severe as the other dystroglycanopathy fish that we have studied in the past, but we were able to show, and you can see it in the central bar graph in the fourth bar, we made the mutation in the full-length mRNA of this early truncation, and we basically saw that it was a complete loss of function, but also it didn't generate any gain of function when we transfected it, when we injected it on its own. And so this is really a complete loss of function that points out that there can be different phenotypes resulting from complete loss of function of POMK, and so that the genotype-phenotype correlation is not direct. So what I have told you so far is that by using this approach of next-generation sequencing and zebrafish validation, we were able to identify 3 new disease genes that regulate alpha-dystroglycan glycosylation. From work of several groups looking at the function of these genes, and in particular from Kevin Campbell at the University of Iowa, people identified the function of these proteins and actually showed that they are necessary for laminin binding. As I showed you at the very beginning, laminin is one of the ligands in the extracellular matrix binding to dystroglycan. There is now a very defined laminin binding glycan combination, and these genes are critical for the formation of that particular structure. And so some of the classical genes that are involved in dystroglycanopathies are POMT1 and POMT2, which add the initial mannose on its chain. And then we found that POMGNT2 would add the second sugar, B3-galin2, the third sugar, on this 3 saccharide, which is necessary for laminin binding. POMK is predicted to phosphorylate this initial mannose, and its structure has been resolved this year, showing that it's a non-traditional kinase that can catalyze phosphorylation of sugar. This event is really important for the addition of this glycan, which is it's a very long, complex disaccharide that is important, again, for laminin binding. So we still don't know how loss of the trisaccharide causes Walker-Warburg this trisaccharide causes Walker Werber, but how the side chain is involved in LGMD and CMD, and this is going to be something really interesting that we and other groups will continue to work on in the future. But now we have all of the zebrafish models, and we're collecting more and more mutations that we can analyze in these models to be able to see what happens. So I'll just give you this is an intermediate summary, and then I'll spend the last 10 minutes telling you a news story. But basically what I told you so far is that we can use homozygosity mapping and whole exome sequencing in consanguineous families very easily to identify disease-causing mutations in these very rare and severe disorders where you may need to focus on only one family at a time. Whole exome sequencing is shown to be very efficient even in non-consanguineous unions, as long as you have multiple affected children. And then you can use the zebrafish as a method of rapid validation for candidate mutations, and you can look at the different phenotypes that are comparable to the ones that you see in the humans. This particular framework doesn't necessarily just fit with dystroglycanopathies. It's now being used across the world for multiple different disorders, and not only neurodevelopmental disorders, because of the rapidity of using the exome sequencing and the zebrafish. With this method, we identified G2, B3-galin-2-K as genes for dystroglycanopathies. And now we're trying to push this approach to tackle sporadic cases. And what I want to tell you in the last few minutes is actually one story where we did that, where we started to look at sporadic cases that had a somewhat different phenotype, a somewhat strange phenotype, trying to find new genes for new disorders. And these are two more people in my lab, Nat Colley, who's a grad student, and Khalid Mushraf, who's a technician, that are working really hard on this project and have helped get it started. So this started from two families that we received from Italy. It was one girl which had short stature, cataracts, and intellectual disability, and one boy with short stature, brain atrophy, and ID. They were not even that similar, but because of the extreme heterogeneity, we now sequence everybody, and we are completely agnostic on what the genetic cause for the disease is going to be. What we knew is that both of them had dystrophic features in their muscle, and that both of them had a disruption in the glycosylation of dystroglycan. And so if you see at the top in the alpha-dystroglycan column, the nice localization of the dystroglycan around the myofibers, and then the complete disruption in the patients, which is not a loss, because we can see there is still dystroglycan present. It just seemed to be completely disorganized and maybe in some cases trapped inside the cell. But this still was presented to us as a dystroglycanopathy, and so when we sequenced these families, we found maybe 3 or 4 possible candidate genes in each of them, but both had mutations in this gene that's called IMPP5K. The girl had 2 heterozygous mutations, and the boy had a homozygous mutation in one of the same alleles that the girl had. So we had basically 3 alleles in this Val23 and 1 in this other location. So this was interesting, and it became even more interesting when we started reading up about IMPP5K. So IMPP5K is a phosphatase, and so which means it removes a phosphate. But it's a very different gene from the ones that we've been studying so far. It's an inositol phosphatase, and what it does, it removes a phosphate in position 5 on both PIP3 and PIP2. So it would act on this PIP2 and this PIP3. So you have probably heard one of the most famous inositol phosphatases is P10. P10 has been involved in autism, it's involved in macrocephaly, and I think it's commonly discussed as a really strong regulator of the mTOR pathway. And so P10 is a 3 phosphatase. IMPP5K is a 5-phosphatase, so it removes the phosphate at a different position on the inositol ring. What these do, they regulate a slew of pathways and of different processes within the cell, including actin regulation, vesicular trafficking, calcium signaling, several signaling processes including mTOR, GSK3-beta, and NF-κB, which lead to a variety of changes and of function within the cell. So this is definitely a really important process and a really important protein. Another thing that was really interesting to us is that IMPP5K was originally identified as SKIP, skeletal muscle and kidney-enriched inositol phosphatase. So the skeletal muscle there being the operative word. It's very important in the skeletal muscle for insulin processing and insulin and response to insulin, and then activation of the mTOR pathway downstream of signaling, of insulin signaling. So we were really interested because we thought it was critical for muscle development, and we had no idea what it could do in the brain. The problem is that we only had 2 cases. And so what we really wanted to do was to find more. And this is where we went to Gene Matcher. So this is the entry page, and this is the URL for this website. This is an effort that all the different genetic centers are running to make scientists work together and to help scientists identify new cases. There are multiple data sharing websites that I want to share with you. There's the Beacon Network, which is global, MyGene2, which is similar to Gene Matcher, and they're all being connected into the Matchmaker Exchange. So the Matchmaker Exchange is a huge effort to basically connect all of these websites that are collecting genetic and clinical information so that both clinicians and geneticists can search these websites for their genes of interest, and they can be connected to each other. And so the way Gene Matcher works is that you express interest in a gene. Every time someone else expresses interest in a gene, you receive an email that says, this person wants to know about IMPP5K, do you want to talk to them? And that's how you connect to people. Also, what I want to bring up is that this is not just for geneticists. I have had basic scientists reach out to me about IMPP5K because they were working on that particular protein. And so if you're working on a mechanism of disease, or you're even working on a basic mechanism, this is a great resource because you can connect both to clinicians and to geneticists that may be working on your gene of interest, and you may find something new about its function, and you may also be able to find great collaborators, as we did. So through this process, we identified 2 additional families that had mutations in IMPP5K. These mutations are distributed mostly in the phosphatase domain and in the ruffle targeting or skitch domain. They are all incredibly conserved all the way to the zebrafish, and some of them are in very, very strongly conserved domain, which are critical for phosphatase function. When we tested the enzyme function in collaboration with Laura Swan's group in the UK, we saw that that most of our mutations very severely reduced enzyme function in IMPP5K in its ability to remove phosphate from PIP2 to PIP1. Through Gene Matcher, we also identified another group that had 12 additional cases with IMPP5K mutations with 4 different alleles. And what this led to, it led actually to 2 back-to-back publications looking at the function of these genes that just came out this month, where we identified basically a new syndrome. We showed that IMPP5K mutations causes syndrome with short stature, muscular dystrophy, intellectual disability, and congenital cataracts, which could overlap with the dystroglycanopathies and also with another known syndrome, which is Marinescu-Jorgen. And we have now 8 different mutations across 2 groups that cause this disease, so we are very sure that this gene is responsible. Just a very quick analysis of the zebrafish. This was interesting because IMPP5K is duplicated in the zebrafish. The zebrafish underwent a duplication, like a genomic duplication event, which caused several genes to now have 2 copies. So we knocked down IMPP5K-A, B, and AB. In general, we found defects in the body length, head length, and eye diameter, only in A and AB together, and not in the B. And one of the reasons for this is that A is the most highly expressed gene, while B is expressed at very, very low levels. This is normal, as when you have duplications, usually what happens is that one of the duplicated copies is silenced so that you don't have twice as much enzyme function, or the other gene starts doing something completely different. But in this case, it looks like it's mostly silenced and non-functional, and we're looking into that. The muscle in the morphine was disrupted. We looked at the ultrastructure of the muscle, and we saw disruption in the sarcomere, disruption in the myocepta, which are those areas where you have the connection between the different muscles in where the extracellular matrix deposits, and the disruption in the triads, which is where regulation of contraction is controlled. We have a smaller head with disorganized eyes, and we also saw a disruption in crystalline organization in the lens during development, which is similar to what happens in congenital cataracts. So again, in this zebrafish model, we were able to recapitulate all of the characteristics of the patients with the myopathy, the small brain, the small body, and the ocular deficits. So what we really want to find out now is, like, how does loss of AMPP5K cause the disease? We know that it's important for AKT-mTOR signaling in the muscle, so that could also be important for the brain, as AKT and mTOR have been involved in multiple neurodevelopmental disorders and definitely cause cognitive deficits. The other thing that's interesting is that SCIP, IMPP5K, is important, is actually transported from the ER to the plasma membrane via GRP78. GRP78 is a chaperone that resides in the ER and controls protein trafficking. This translocation is critical for SCIP's regulation of signaling, but also, since dystroglycan is glycosylated and its transport is regulated in the ER, we wonder whether this could be the origin, like this lacking of this interaction could be the origin for the disruption in dystroglycan expression, and is something that we're definitely following up. And so, to conclude, what I told you today is that rare neuromuscular disorders and neurodevelopmental disorders affecting brain function and development are very genetically and clinically heterogeneous, but that we can use these techniques to really make gene identification much faster when we can use next-generation sequencing, and then an animal model like the zebrafish, which allows you to look at differentiation of multiple organs in vivo very quickly. I also hope that I convinced you that sharing genetic and clinical information has been going on in our community, and it's incredibly important. We would not have been able to complete this last story if we hadn't met so many different groups through GeneMatcher and through these matching services, and all started working together to combine our patients, to combine our expertise, and to push these papers through publication very quickly. And then the last point that I want you to take away is that, yes, dystroglycanopathies are mostly caused by mutations in glycosyltransferases, but this latest IMPP5K mutation identification raises the possibility that there may be also additional mechanisms that are possible. And so I want to thank everybody in my lab and our wonderful collaborators, the sequencing centers that have helped us with the sequencing, and the collaborators that have provided the patients we're working with. I thank the patient families and our funding sources. And I will take questions.
Thank you for that informative presentation, Dr. Manzini. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, how many genes do you think we are still missing for the remaining 50% of cases?
That's a great question. So the problem is that, as you have seen, we are focusing on the coding regions, and the vast majority of what we're doing is in the exome and in the coding regions. We are really not considering yet, or we're starting to consider now, non-coding and regulatory mutations. It seems that this 50% threshold is very hard to break. So we keep finding new genes, but they only affect a very, very small number of patients. So there is nothing that is more common that we're missing, we think. So it's either there are another 20 genes that cause very, very rare forms of the disease, or it's absolutely possible that the genes we already know are responsible for a large portion of the cases through non-coding mutations that are either intronic or in the promoter or in enhancers that regulates the function of these genes. And that really, the understanding the non-coding genome is really the next frontier, and I think it's going to be very interesting.
Thank you. For our next question, is the zebrafish sensitive enough to model disease variability? I think it's a good question. It definitely depends on the assay and how well your rescue works. We have had a lot of success with that, with some genes where we were actually able to model moderate versus severe presentation, recapitulating that in the zebrafish, where we could show that a mutation that causes a very severe presentation and complete loss of function causes a more severe phenotype in the fish, and a mutation that only partially affects the function of the protein can rescue to a certain extent. That completely depends on how good your rescue is and how good your morpholino is. Thank you. Our next question is, are morpholina oligonucleotides really the best approach to model loss of function? I heard they have a lot of issues.
Oh, there I am. There's a huge discussion in the field on whether we should use morpholinos at all. I personally think that they get a bad rep because people sometimes are not careful on how they use morpholinos. You have to be very careful. Some of them are bad, and you have to be able to distinguish when a morpholino is bad and when it's having a specific effect and when it's not. What we usually do, we want to use the lowest possible concentration. We want to make sure that it's rescued by the human mRNA, and that the function is conserved, and that the gene that you're removing is actually the gene you're interested in. What we're also doing in parallel, we're using CRISPRs. And so I think nowadays you need to have multiple validation methods to really show that the phenotype that you're getting is the correct one. We are certainly heartened by the fact that we can recapitulate the human phenotype, and so that we have a specific set of molecular changes that we see in the human, such as the loss of glycosylation, that we also see in the zebrafish caused by our morpholinos. But yeah, there is definitely a lot of discussion in the field on which is the best method to use and which is the most valid.
Okay, we have time for one more question. How would you recommend a cell biologist use the matching websites that you discussed?
Yeah, that was very cool, because when I started getting requests from cell biology labs that do genetics, I was like, why are you even on this website? But then I realized it's actually a great tool. If you are working on a gene that you think is related to disease, or it's related to a gene that causes disease, by demonstrating interest, you can be put in touch with other groups for collaboration. And so now I'm actually really, I'm starting to think more from the aspect of the cell biology that we do in the lab, and start expressing interest to a lot more genes. Because you can identify people you can work with, you can identify people that do particular biochemical or biological tests that you want to use. I think it just in general is going to speed up the process of discovery. And for us, it was really transformative, because it brought a mutation that had been identified for almost 3 years, where we really didn't know what to do with those 2 patients, to having 2 publications in the range of like 4 months. Because we all came together at the same time, we were all ready to move forward, and it was a really excellent experience.
I would like to once again thank Dr. Manzini for her presentation. Do you have any final comments?
No, I would like to thank the crowd and everybody for the questions and for listening. So this was wonderful. I'll send a shout out to Dr. Yalda Jamshidi, who really was my partner in crime for this latest AMPP5K project, because I realized I didn't single her out as I was going through the things very quickly. But without collaborators, really, we wouldn't be able to do everything that we do. Thank you.
Thank you, Dr. Manzini. Today's webcast will be available for on-demand viewing through June of 2017. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.